So I'm very pleased to introduce Dr. Natalie Gilmore as our speaker for today's Rehabilitation Science seminar. Dr. Gilmore received her BA from the University of Pittsburgh and her master's in medical speech-language pathology from the University of Washington. And prior to pursuing her PhD with Swati Kiran at the University of Boston, Dr. Gilmore worked here at Moss Rehab as an outpatient SLP. So hopefully she is still familiar to many of you. And during her time here, she also volunteered with Dr. Myrna Schwartz with us here at MRI. Currently, she is a research fellow at the Laboratory of Neuroimaging and of Coma and Consciousness at Massachusetts General Hospital. And her research program is centered on theoretically grounded intensive rehabilitation programs, studying them to see how they facilitate recovery of language and cognitive function after brain injury. Today, we will have the opportunity to learn about some of that work. And with that, I just want to thank you very much one more time for accepting our invitation to present today. And we look forward to learning from you.
Thank you so much, Maria-Lisa. It's a pleasure to be here. I really appreciate the kind introduction and again, the invitation. So I will go ahead and get started because I do have a lot of slides and I will try to leave time for questions as well. Okay, but if we can save them until the end, that would be great. Okay. All right. So good afternoon, everyone. Thanks again for coming today. And it's great to be back at Moss to share this work. So today I'll be presenting again about intensive cognitive and communication rehabilitation for young adults with acquired brain injury. And this is work that I conducted as a part of my doctoral studies with Swati Kiran at the Boston University Aphasia Research Laboratory. So again, this is part of the Shrier Family Visiting Scholar Lecture, and this is supported by a gift from Nancy and Marc Shrier, and we're grateful for that. And again, I don't have any financial relationships to disclose, and neither do the planning committee members in terms of the work that I'm presenting today. And just some brief learning objectives. I'm hoping you're going to be able to recognize some evidence-based approaches to cog rehab, principles of experience-dependent neuroplasticity that are relevant for brain injury rehab, and then also some of the key components to the intensive cognitive communication rehabilitation program that I'm going to be talking about today. So you may have had a sense of this from the title and learning objectives on the last slide, but I'm going to go ahead and just provide some, in terms of what we're going to talk about, we'll do a background on acquired brain injury or ABI, cognitive rehabilitation, the needs and the state of rehabilitation for young adults who sustain ABI. Again, the program that I keep talking about, intensive cognitive and communication rehabilitation, I'm going to refer to that as ICCR in the talk going forward. So just for anytime you hear those letters together. And then also I'd like to kind of transition from talking about behavior to neuroimaging and how we can use functional near-infrared spectroscopy or Effner to study language and other cognitive processes in healthy adults and also folks with aphasia after stroke, and then potentially how we could use it to measure changes in the brain after intervention in a few young adults with ABI that we piloted this approach on. And with that, I'll just go ahead and get started. So as many of you in this who are with us today are probably aware, acquired brain injury or ABI refers to brain injury that occurs after birth that is not due to congenital, developmental, or neurodegenerative causes. It is an umbrella term. It encompasses a range of etiologies, with stroke and TBI being most common, and roughly about 800,000 individuals experience stroke per year and 3 million sustaining TBI on an annual basis here in the US, with young adults being a relatively frequent and/or growing population to sustain ABI. And this is particularly important for the talk today. So in order to understand the impact of these different injuries on function and behavior, it can be really helpful to think about whether the injury was focal or diffuse, or whether it was primary or secondary in nature. Stroke is a classic example of focal primary injury and often has the most impact on function of the area in the area of infarct, but it can also affect more remote areas from the lesion. Diffuse axonal injury is a hallmark example of diffuse primary injury with very widespread effects on function. And then again, there's always these secondary injury processes that we need to consider as well that occur in response to the primary injury, and whether that's focal or diffuse in nature, and it can exacerbate its effects by leading to additional neuronal damage. So ABI being a very complex injury. So those mechanisms that I was just talking about, they lead to cognitive impairment, right? So there are deficits in attention and memory and executive function, and these are all common in traumatic brain injury, tumor, encephalitis, and anoxic hypoxic injury. Language, their visual spatial constructional impairments are seen across etiologies, whether that's left hemisphere stroke or TBI. Language impairment we often think of as being most often associated with left hemisphere stroke. But individuals with TBI, tumor, et cetera, can also experience language and other types of communication deficits after injury. And the same is true for individuals with left hemisphere stroke. Some of those individuals can also show, beyond aphasia, they can show impairments in attention, memory, and executive function. So overall, individuals with ABI do appear to have can have overlapping patterns of deficits in a range of cognitive processes. And that's important for the program I'm going to talk to you about today. So now that we kind of have a sense of the brain being damaged after ABI and how that impacts cognitive functioning, let's think about what processes or factors may actually drive recovery after ABI. So there are a host of interrelated biological processes that occur at the molecular and cellular level to support recovery, such as neurotrophins or growth factors that support the survival of brain cells or neurons, synaptogenesis, or the creation of new connections between neurons that form new neural pathways, and also this can then facilitate brain reorganization. There are individual specific factors. For example, there's research that's shown that, maybe individuals who are younger may adapt faster than older individuals after brain injury. There are also studies suggesting that individuals with very positive that have higher social support may have more positive outcomes from rehabilitation. And then, we're really just starting to understand or actually try to study and measure how intrinsic factors like motivation impact recovery. There are also behavioral drivers of recovery, like cognitive rehabilitation and principles of experience-dependent neuroplasticity. And these represent the notion, again, when I talk about principles of experience-dependent neuroplasticity, I'm thinking about how the brain adapts, either in the case of normal learning or recovery of function in a damaged brain through cognitive, sensory, and behavioral experiences. And we'll drill down into each of these factors a bit more. So let's start off with cognitive rehabilitation. And I think that this topic is very familiar to many of you in the audience. It's the gold standard of care for targeting cognitive impairments that are experienced by individuals with ABI. It's provided by a range of professionals in a variety of settings and through a spectrum of approaches. It can focus in on just a single cognitive process or multiple cognitive processes at the same time. And these cognitive rehab programs they're naturally sort of embedded with principles of experience-dependent neuroplasticity, suggesting that they really should promote changes in the brain or changes in behavior and then in the brain. And we should think about this as recovery, right? Nevertheless, none of these approaches are really without limitation. So maybe a reasonable step might be to think about how we could combine different advantageous aspects of each of these approaches into one intervention that could then maximize outcomes for ABI. And so what would that look like? That integrated approach, perhaps we would try to target impaired processes that aligns with maybe a restorative approach to cognitive rehab. And that would be in agreement with principles of experience-dependent neuroplasticity, such as use it or lose it, use it and improve it, or specificity. We may also want to minimize reliance on spared processes. And that may vary depending on like how far post-injury individuals may be. But in this case, we would maybe limit at certain times in the recovery phase, we may limit compensatory approaches to targeting cognition to avoid interference. We also want to provide many opportunities for rehearsal or practice, which aligns with this comprehensive kind of approach to CR. And that would be in agreement with repetition mattering and intensity matters. And then again, that the rehab should occur in a variety of contexts, which would encourage transfer of learning. And this is maybe in agreement with the contextualized approach to CR. And this would align with principles of salience, matters, and transference when we're thinking about those principles of neuroplasticity. So with that framework in mind, I think if we turn our attention to the young adult population that I mentioned earlier on, and we think about the impact of ABI in this group, the rate of stroke has been increasing in young adults in the last several decades, and this has been attributed to some extent to an increase in vascular risk factors in this population. Young adults are a frequently affected age group to sustain TBI, and we know this in relation to sports or recreation-related injuries, motor vehicle accidents, falls, assaults, other common causes of TBI. The opioid epidemic in the U.S. has been has led to an increase in non-fatal overdoses and also an increase in hypoxic, anoxic injuries. And just when we think about the different types of injuries that I'm mentioning here, when you sustain an ABI at a young age, it can be particularly devastating. It can lead to reduced academic and work participation. College in particular, which we think of in the US as a major developmental milestone, providing opportunities for us to increase our independence from our parents or form lasting social relationships and advance toward a lifelong career. This can often be disrupted. This pathway to college or a career is often disrupted for young adults who sustain ABI. And then overall, their studies have shown that young adults with ABI do endorse reduced satisfaction and overall quality of life post-injury. And this could be in part due to some of the loss of some of these experiences that they're not able to take part in the same frequency or at all. So continuing on from this previous slide, ABI makes college difficult. It impacts cognitive processes that are important for academic success. Students rely on their attention so that they can complete many different activities in college, like listening to a lecture. Memory is really important for recalling information when taking a quiz, for example. Students also, maybe they need to plan their day, organize how they're going to get all of their work done. Again, these are executive processes, right? They rely on language very heavily to give a presentation, to read a textbook, to write an essay, amongst a host of other things. And then, visual spatial processing, just as one small example, it could be recruited for many tasks, but specifically maybe pathfinding on a college campus could be particularly hard for some individuals due to challenges to this domain or deficits in this domain. And the impact of ABI on cognitive processes important for academic success is further impacted by the current gap in the continuum of care for services to advance these young folks with language and other cognitive deficits after ABI to college. Existing programs, incredibly valuable, and overall the course of recovery, though, does not necessarily appear to be sufficiently repetitive, intensive, specific, salient, or even appropriately situated within an academic context to support return to academics. And we know this to be true given the struggles that individuals with ABI continue to report, that they're having a difficult time returning to school post-injury. Some comprehensive cognitive rehabilitation programs have supported return to school, but looking at the literature can be really difficult to discern how many of those individuals actually went to college from such programs, because the outcomes are often conflated in terms of just overall productivity and return to work and school being combined into one outcome. And then beyond that, understanding how cognitive function, what the cognitive outcomes have been from such programs is also has not necessarily been consistently or thoroughly assessed as a program outcome measure. And these are really both essential steps in evaluating the utility of such programs and preparing young adults with ABI to transition to college. And so one thing that we did at Boston University over the time that I was there was we developed the Intensive Cognitive and Communication Rehabilitation Program as one potential way to address this gap in care. So this is a simulated college experience. Approximately 6 to 8 young adults with ABI would attend classroom style lectures, individual therapy, and application kind of computer-based training, 6 hours a day 4 days a week for at least one 12-week semester, or until they were ready to transition to college. And I'm going to kind of dive into each aspect of this program in more detail across the next slides for some of the clinicians who might be in the audience today. So as part of the core courses that you saw, that kind of like 10 to 1 p.m. block, the participants would, and these were core courses that would alternate in topic, either Monday, Thursday, or Tuesday, Friday. Participants would watch a pre-recorded video lecture, usually something like Yale Open Courses, where they're quizzed on each course weekly. They would also, this is a time when they would be able to practice strategies to maintain attention. So maybe they would want to stand up briefly, or they would, work on jotting something down to refocus to the lecture. After they'd watch the video, they would review the material as a group. The speech pathologist in the room would encourage everyone to share something that they recalled from the lecture that they found to be particularly important. We would call this brain dump. They could then use notes that were provided to them at the start of the lecture to support their recall and word retrieval, because we knew that for some individuals it was particularly challenging unless they could kind of re-access some of the information from paper form. And this is also a time where they could engage in kind of metacognitive strategy usage. So maybe they would recognize that they didn't recall something and that was something they were going to need to study more later, or they could generate associations or mnemonics to recall certain concepts. And this was something that they could repeat on a daily basis. This is 4 times a week across a 12-week semester with different content each day. So after that lecture review session, they would go through their practice quiz questions on the content they reviewed as a group. They would see, everyone would see a question on the slide, and then they would write down their answer. And then folks could share their responses. They could explain why something was accurate or inaccurate to one another. This would really provide opportunity for them to see what they needed to study more later again, to mark something in their notes that, oh, I didn't understand this. And then also just get some practice using test-taking strategies like eliminating implausible answer choices, et cetera. And these were all things that, again, were able to be reinforced on a daily basis. And then in real time when they would take an actual quiz on this content the following week. So we already kind of touched on metacognitive strategy instruction. This is just one example of a strategy that the students used, something called RITA, which stands for rehearse, imagine, take time, and activate. And this was a way to really encourage the students to engage in the lecture content more deeply and encourage encoding and later retrieval. And you can see some others on the slide here. So after lunch from 12 to 1, they would have individual therapy or an elective course. And ICCR really included a range of topics for the elective courses. These courses were small group or seminar based in style. For the English course, for example, the students would read a chapter or 2 of a novel, and then they would come to class prepared to discuss that and answer specific questions about the content. We would give them a summary of the chapter that was freely available online, and then they could work on highlighting and underlining and starring key information to support their comprehension and recall of the information later. We've also done a public speaking course where they'd prepare presentations. In this case, maybe someone would pick a specific skill that they were working on, maybe speaking slowly and clearly, and then they would give themselves feedback on how they did, and then they would also receive feedback from their peers. And so really nice opportunity for them to work on public speaking in a safe space. And then beyond that, it was an elective course because they may actually miss it for individual therapy, as opposed to those core courses, the 10 to 1 block. No one was ever pulled out of those courses for therapy, but this elective course, we would alternate so that nobody missed it too often, but that occasionally folks would. And they were still quizzed on this content as well. So this was, again, so they were actually getting 4 weekly quizzes, a quiz each course, to really encourage them to be accountable for the information that they were showing up to at the lectures. Okay, so during that last hour of the day from 3 to 4, they would attend a technology time. This is where they would work on specific language and other cognitive processes using constant therapy, or they could access notes from the ICCR course website. This was sort of developed to replicate a Blackboard or Canvas type experience that most college students are familiar with, where they're locating assignment notes or they're looking at a course syllabus. And then also they had Google Suite essentially, so that they could work on using Word and PowerPoint and how could they dictate, and kind of getting comfortable with using different accessibility features of their laptop or iPad for completing their academic assignments. And they could do it in a supported environment with the speech pathologist there. And then I did mention that they receive individual therapy. You can just see a couple of different images on this slide. Essentially, the individual therapy targeted their needs based on their personal goals, things that we noticed that they were struggling with in the classroom, and then also domains of weakness or identified from standardized assessments that we administered before and after the program. And so the SLP would generate a treatment plan with them at the start of the semester, and then implement a variety of impairment and functionally based approaches to address those goals. So the program also included a lunch and learn seminar to support transition from ICCR to college. So once a week, students would hear from speakers. They could be individuals who graduated the program. It could be people working in disability services, an occupational therapist who would guide them on living independently, just a range of different topics depending on what the students were interested in, what they needed, and what we thought would support them. And they would also work on things like, managing time as a student, what disability and academic resources were available on campus, how to access neuropsych testing for accommodations, et cetera. So a really nice opportunity for them to kind of work toward transitioning. Okay, so now that I've kind of given you, we've looked at the program as a whole, we've dug into some more finer details of the program, and I hope that I've been kind of, if we go back to this framework that I presented before of kind of what a revised integrated COG rehab approach might look like, I hope I've built some of a case that the ICCR program aligns with this revised integrated approach to CR. For example, in this ICCR program, individuals with impaired writing that was based on a standardized assessment they would target spelling in their individual therapy, and then they would also get to write on a daily basis, which would be in line with restorative CR approaches and principles of use it or lose it. They're getting to write all 4 days of the week in multiple contexts. They would also get to write notes daily in class and work on essays in their seminar, which is in line with the comprehensive CR approach, with these daily sessions of group work in line with principles of repetition and intensity matters. And then finally, that they would also work on writing a position essay about a topic that they're really passionate about in line with the principle of salience. And then they'd also be given homework to write a college essay to encourage transfer of learning in line with the contextualized CR approaches that encourage therapy in a variety of contexts. Okay. So at this point, I think that, I hope that I've made some case that the assessing the extent to which an intensive integrated cognitive rehabilitation program like ICCR promotes gains in cognitive function is essential intermediate step for us to evaluate how such programs do, how successfully they actually prepare young adults with ABI to advance to college. And then starting to examine how the brain is changing in parallel with these cognitive gains through neuroimaging could maybe provide a mechanistic understanding of the intervention. And so let me kind of dig into the neuroimaging research a bit here before we dive into the studies. So to date, most neuroimaging treatment studies in ABI have applied task-based functional magnetic resonance imaging, or Fmri, with mixed findings. Some studies showing increases and others showing decreases in post-treatment activation in conjunction with behavioral gains. Fmri can be contraindicated in the ABI population. So, for example, maybe due to ferrous material, although that's less common nowadays, but also just challenges lying flat, sensitivity to noise, whereas functional near-infrared spectroscopy, or FNIRS, can circumvent some of these challenges. It's safe for ferrous material and medical implants like programmable shunts that many of our folks with ABI have. And it can also be conducted in a natural environment. It's very quiet. You can see here, this is one of my former lab members who's wearing a cap on her head. It positions the fibers on the scalp that are connected to the measurement device that you can see in the background on that cart. And these will allow us to measure neural activity. And I'll tell you a little bit more about how that happens in the next slide for folks who aren't as familiar with FNIRS. So FNIRS relies on optical properties of hemoglobin rather than magnetic properties like Fmri. So short sources will shine light through brain tissue at multiple wavelengths, and then any light that's not absorbed or scattered is captured then by detectors at the scalp. And you can kind of see that the photons travel in a banana shape here. And so that's what's being captured in order to measure the concentration changes in hemoglobin that can be estimated using something called the modified Beer-Lambert law, which if anybody really wants to go into it, we can talk about it afterward. So these calculations will provide a thorough estimate of the hemodynamic response, or HRF, with information about the oxygenated hemoglobin in red that you can see here, and the deoxygenated hemoglobin in blue here, and then also total in green. And so this shape that I'm pointing to over here in the graph is essentially the canonical hemodynamic response to a stimulus that's evoked when there's an increase in HBO or oxygenated blood and a decrease in deoxygenated blood. And you're going to see a little bit about that in study 3 when I get there. Okay, so numerous studies have applied Effners to study language and other cognitive processes in healthy adults. This is a topic of interest in this work is in terms of the engagement of left lateralized language network during semantic processing and lexical retrieval, and the recruitment of the domain general network during hard versus easy arithmetic processing. And this is, there have been 2 published studies that have applied Effners to study language processing in individuals with aphasia, one at a single time point and one before and after a combined neurostimulation and language therapy study. Nevertheless, strong conclusions from these studies are challenged by some methodological limitations. They didn't necessarily cover the full language network, maybe only focusing on, superior temporal gyrus or middle temporal gyrus. They didn't necessarily compare to an active control condition, so maybe not a very specific contrast for understanding language processing. And then also just kind of limited description of the methods that were used, which makes replicability challenging, and so suggests that we need to kind of keep doing this work to better understand how it can be applied in the damaged brain to study language and other cognitive processes. So all that being said, so based on everything I've shown you so far today, I'm hoping that it's clear that, there are limited cog rehab services to advance young adults with ABI to college. Some cog rehab programs have targeted return to school, but cognitive outcomes have not always been consistently or thoroughly assessed. Prior studies that have used Effners to examine language and other cognitive processing in neurotypicals and individuals with ABI, there are prior studies that show it to be feasible, and there are advantages to applying such techniques to assess treatment-related neuroplasticity after cog rehab in ABI. And so in this work, the focus, kind of thinking about all of this research, had spurred the development of this kind of overarching hypothesis that I'm going to kind of try to present to you today, which is that cognitive rehabilitation programs that are embedded with principles of experience-dependent neuroplasticity should promote changes in behavior and changes in the brain in young adults with ABI, and this can be captured using Effners during language and domain general processing. So let's talk about the studies that we used to try to examine this, to test this hypothesis. So let me this is redundant, so I'll just move right into study 1. So starting with study 1, the overall aim of this study was to determine the initial efficacy of ICCR by measuring whether participants demonstrated changes in cognitive linguistic function, classroom participation, therapy, and their personal goals, and also their participation in quality of life. You can briefly see the participants here. We had 4 folks who participated in the treatment and 2 who participated as deferred treatment usual care controls, meaning that they could participate in their everyday lives, they could volunteer, they could go to therapy, anything other than taking college courses or doing another intensive program. Everybody's between the ages of 21 and 34. They're all in the chronic phase of recovery. They've all finished high school, and they showed some language and or cognitive impairment as evidenced by the WAB, or the Western Aphasia Battery, or the repeatable battery for the assessment of neuropsychological status, or the RBANS. So before and after each 12-week semester, so I mentioned that the program's, it's 6 hours a day, 4 times a week for 12-week semesters. They also did some standardized testing. I've already told you a little bit about the Western Aphasia Battery and the R bands, but they also did something called the SCAN, which is the Scales of Cognitive and Communicative Ability for Neurorehabilitation. And it measures more of a range of language and other cognitive domains like attention and reading and problem solving. And then also the discourse comprehension test, which looks at auditory comprehension and reading comprehension at the paragraph level. Beyond these standardized assessments before and after each semester, they underwent measures of participation, like the Child and Adolescent Scale of Participation, measures of quality of life, like the TBI-QoL and NeuroQoL, anxiety, positive affect, short forms, et cetera. And then we also did some goal attainment scaling with them, in addition to measuring their participation in the classroom, scoring essays, scoring their quizzes, and their individual therapy goals. So they engaged in the treatment, which I've reviewed earlier, so I'll just kind of breeze past that. So in this first study, remember, it's a pretty small study. This is after our first semester of trying it. We weren't even sure if we could get this kind of program off the ground. And what did we find? So we find at the group level, these are linear mixed effects models, essentially using time point as a predictor of score. So does score go up as time in the program goes up? And what we find is that on the Western Aphasia Battery and the SCAN, individuals' scores significantly increased the longer they were in the treatment program. And then at the individual level, treatment participants showed significant gains on at least one standardized assessment, whereas control participants did not. And this provided some early support that the gains that we were seeing in the treatment participants were attributable to the intervention, just again, early support. In terms of their classroom participation, all the participants who were in the treatment, they showed that over time, there was a significant increase in the positive behaviors. So things like asking appropriate questions, making appropriate comments to the lecture, et cetera, increased at a faster rate than the negative behaviors, which would be things like asking inappropriate questions or making inappropriate or irrelevant comments, which is pretty exciting. And finally, they did increase in the complexity of their individual therapy goals. The majority of them showed an increase in their life participation and also their quality of life. And then I'll get into this in more detail in a future study. But again, this was all in all positive findings for the first kind of early study that we put out and related to this work or related to this program. On study 1, so building on study 1 with more participants, we moved into study 2. So we wanted to assess the effect of ICCR on specific cognitive domains over time in young adults with ABI. So before we were kind of looking at these big gross measures, the Western Aphasia Battery, aphasia quotient, which is a big kind of composite of language function. And, the R band's total score is a composite of all of these different cognitive subdomains. So we wanted to try to drill down into more specific language and other cognitive processes. So you're interested in whether there was a significant difference in overall item accuracy between treatment and deferred treatment control participants over time. So essentially, was there a big overall effective treatment? And then we also wanted to look to see if within those specific subdomains like attention or memory or problem solving, did we see a significant difference in subdomain item accuracy between the treatment and the deferred treatment control participants over time? So here you can see we were able to increase our sample size a bit to 22 in the treatment and 14 in the deferred treatment control group, with a relatively even split of etiology, with half of them in the non-traumatic brain injury group, like stroke, tumor, encephalitis, and the other half with traumatic brain injury. Everybody had completed high school at the time of enrollment. And similarly to the previous study, they had demonstrated language and or other cognitive impairment as evidenced by the WAB-AQ or the R bands total. Scores below normal limits, either 93.8 or below at least one standard deviation from the R bands total of 100. So a score lower than 85. And all the participants in the treatment group tended to contribute at least 3 time points of data, and individuals in the control group contributed at least 2 time points of data to the study. So this was, again, cumulative effective intervention. They they all did the same standardized assessment battery that I showed you about in the previous study, and they had the same eligibility criteria. Rather than looking at that effective intervention using these big traditional aggregate standardized assessments, as we did in study 1, we took items from the standardized assessments and we assigned them to 10 either language or other cognitive domains, as you can see here. And for example, we took all the items in the auditory comprehension sections of the WAB, all the items from the speech comprehension subtest of the SCAN, and all the items from the listening version of the DCT, and we assigned them to the domain of auditory comprehension. So that's what I mean by we took these big tests and we broke them up into domains, and we used traditional neuropsych reference materials to do so, like Lezak's 2012 neuropsychological assessment manual, reference book rather. Okay, so then in terms of data analysis, these were all analyzed at the between and within group levels. So we had 2 sets of models there. And at a high level, what you need to know is that we were looking at whether item accuracy significantly went up over time across all the items. That's this model. And then whether or not we saw that within specific subdomains. And we did include random factors to account for differences in pretreatment accuracy for participants. So people who were, some people were starting really high on certain domains versus others. We also tried to account for differences in the rate of change across participants, and also different complexity in items. Etiology, so whether folks had TBI or non-traumatic brain injury, was included as a categorical covariate to try to get a sense of whether or not the effect of any effect we saw of the intervention was different for folks with TBI versus non-TBI, as that's a common question. So in terms of the between group analyses, we found that item accuracy for treatment participants improved at a significantly faster rate than the deferred treatment usual care controls over time. So this is really emphasizing that we saw an overall effect of the treatment. And then when we looked at the time point by subdomain by group model results, we were able to see that treatment participants improved at a significantly faster rate in verbal expression. And then we also saw positive findings in written expression and visual spatial constructional domains that did not withstand, multiple comparison correction. Finally, and then we followed this up with an overall, with a 3-way interaction of time point group and etiology, essentially to see, as I was saying before, whether or not the effect of the intervention differed, whether you had TBI or a non-traumatic brain injury, and that difference was not significant, suggesting that the intervention really did have a similar benefit for folks with non-traumatic and traumatic brain injury. And that's really meaningful for young adults with stroke, I think, in that those folks often find, can find it hard to have a peer rehab group, when often spending time with older folks with stroke, and they're, with the mean age of stroke being 60. So I'll dig into that a bit more when we get to the conclusions, but overall positive results here. And then in terms of the within group subdomain model, treatment participants showed significant increases in item accuracy in verbal expression, written expression, memory, and problem solving, and no such gains were seen in the deferred treatment usual care control group over time. Again, providing some early, these are exploratory findings really that are promising in that young adults with ABI showed significant gains in cognitive domains important for academic success. And this did, kind of address one of our primary aims in this study. So to summarize, building on study 1 and study 2, study 2 really provided strong evidence that ICCR promotes gains in overall cognitive function and also in specific cognitive domains important for academic success. And given these positive findings, we thought it was logical to move forward to seeing what's going on in the brain and how that may be supporting some of these gains that we're seeing in behavior. And so for that, I'll move quickly into studies 3 and 4, being mindful of time. So in study 3, our overall aim was to assess the feasibility of Effners to capture brain activation in expected regions in neurotypicals and individuals with post-stroke aphasia during language and domain general cognitive tasks. So we were interested in what brain areas were recruited for language and domain general processing by healthy individuals as measured by Effners, as you see here, and also in domain general processing. And so we had a mix of groups here. So we had a young healthy cohort, an older healthy cohort, and then several individuals with chronic aphasia that we were using as a pilot in this study. And everybody, yes, so I'll just move on from that. I don't want to belabor this too much, but essentially we used a 56 channel probe that covered your bilateral frontal, temporal, and parietal areas of the brain. We had long separation channels as well as 8 short separation channels. The main thing to know about with the short separation is that that measures blood flow at the scalp, and you use that as a regressor to remove any additional noise from the signal that's related to heartbeat, respiration, or blood pressure fluctuations. So again, kind of just improving the precision of our measurement of the hemodynamic response. After each measurement, we got digitized points of where each source and detector or channel was located. And then that allowed us to get estimates for where we were measuring in the brain. So during the actual Effner's measurement, we used traditional task-based, we used traditional tasks that had been previously validated in the aphasia research laboratory with individuals with aphasia using Fmri. So everybody performed 2 runs of each of these 3 tasks. This first one here is the semantic feature task. It was event related. There were 2 conditions. So the real condition, they saw a picture and they had to read the phrase in their head and then press yes or no if the phrase applied to the image. For example, they saw the apple is eaten fresh and they would have to click yes or 1 to indicate yes, that an apple is eaten fresh. In the scrambled condition, they would see, you can see that image there that is like dots with black and white dots, and they would see the phrase color, and in this case, they would press 2 for no, that is not in color. So that's essentially the difference between the real condition and the scrambled condition, with one having to do actually accessing more higher level semantic information or meaning about the item. And then picture naming. So again, 2 conditions. So this was a block design. You can see some more details about the task there. Essentially, the individuals would see a picture and they would have to say the picture aloud. In the scramble condition, they would just say the word skip. And so in this case, we're kind of removing the lexical retrieval part of that task in the scrambled condition. And so hopefully we're getting some sense of what's going on when folks are actually naming the picture accurately. So that is the picture naming task. And then here's the arithmetic task. This was developed off of a task that Ed Fedorenko and colleagues use regularly to assess the multiple demand network. And we were using it here to assess domain general processing in adults with aphasia and healthy young and older adults. And in this case, we have the hard addition task and we have the easy addition task. In either case, they're adding either a double-digit number to a single-digit number or a single digit to another single digit. And essentially, we're just contrasting what's happening when you're having to do something that is more complex or requires more processing. In terms of the Effner's data analysis, you can just briefly see our processing stream here. If anybody else is interested in Effner's, wants to talk about it in more detail, I'm happy to do so. Just let me know. Really quickly, I'll just highlight that we excluded channels with poor signal to noise ratio. We use motion detection and correction methods that are particularly important for the overt picture naming task, in which individuals actually were speaking aloud when naming. And then we also used a flexible general linear model to estimate the shape of the HRF. And this was all done using Homer 2, which is a package available on GitHub. Okay, and then in terms of managing the lesion, so we took some additional steps in the stroke group, the 6 folks with stroke. So we acquired structural scans on them. We drew a lesion map manually to identify where the lesion part of the brain is. We normalized that to M&I space. And then we took the digitized points from where the channels were when they were wearing the cap, and we projected that probe to the cortex. And then that would allow us to identify whether or not any of our channels were actually in an area of frank lesion. If they were, we would turn them off, essentially saying we don't want to look at signal there. And then we would rerun the GLM or the generalized linear model so that those channels were not contributing, potentially spurious data to the analysis. And this is one way that we decided to manage lesion in this study. There are going to be other ways as this field continues to move forward. So all the statistical analyses in this study were for the behavioral data, we used paired sample t-test to look for differences in accuracy and reaction time between the task conditions. So between, the real and the scrambled condition, or the hard and the easy condition. For the neural data, we conducted paired t-tests at each second of the time course to assess for a difference in oxygenated hemoglobin, or HBO, and deoxygenated hemoglobin, or HBr, concentration changes between the task conditions. So essentially looking to see if there was a difference. So how was there a difference in the HRF depending on condition? So in terms of the behavioral data, we see largely the same pattern across all three tasks. So all three groups show higher accuracy and faster reaction time in the controlled and experimental condition. And then you can also see that the younger healthy controls were the most accurate and fastest to respond, and the individuals with aphasia were the least accurate and slowest to respond, as expected. But they were able to consistently perform at least at chance on the task, which shows that they were able to actually participate and they were doing the task. So moving on actually to the neural data, I'm just going to kind of try to breeze through this for everybody without getting into too much detail I'll try to orient you quickly. So you're seeing here, these are mean HBO concentration change maps, which are essentially showing you what is the activation when we take the real condition minus the scrambled condition at a 5 to 10 second range of time, which is when we would actually expect to see a response to the stimulus. The warmer colors show higher HBO concentration change or more oxygenated blood in the area. And then what you're seeing on the time course plots here is essentially HBO concentration change over time, with the real condition in red and then the scrambled condition in this maroon. And so we can see that these here is kind of challenging to see, and it's small on this slide, but essentially all of these figures that I'm showing here, there was increased recruitment of right SFG, left IFG tri, or left inferior frontal triangularis, left inferior frontal gyrus opercularis, and then left middle temporal gyrus during semantic processing in the healthy young adults. And then what we're also going to see in the healthy young adults, the older healthy adults, and then the individuals with stroke here. Okay, and then really quickly in terms of HBr, HBr, the deeper blue means that there's lower mean HBr concentration change for real than scrambled, which again would be consistent with activation during semantic processing. And so what you can see here, so the real condition is in blue and the purple is the scrambled. And so in the young healthy controls, what we saw was increased activation during right middle temporal gyrus, and left inferior frontal gyrus opercularis, and left precentral gyrus. In the older healthy adults, we're seeing it in left inferior frontal gyrus or triangularis, left inferior frontal opercularis. And then in the individuals with stroke, we're seeing left superior frontal gyrus and left supramarginal gyrus. And so overall, seeing this robust engagement of language areas that's more notable in the younger healthy controls, and we're mostly seeing it in the HBO. And we're also seeing, this is pretty consistent with left IFG engagement in older healthy controls, and then our folks with aphasia engaging a little bit of superior frontal and left supramarginal gyrus, which is actually more consistent with, recruitment of areas outside the traditional language network in individuals with aphasia. And that's a pattern that we don't see in the healthy controls. So I think in the interest of time, I'm going to just breeze through this part even more quickly. So what we're going to say here is that essentially this is a picture naming task. Everything is in the same orientation. You're seeing the HBO and then the HBr on the bottom. And in terms of the results, the young healthy adults they're showing higher HBO concentration change for scrambled than real in the left inferior frontal gyrus and triangularis and opercularis. And then we're also seeing that the older folks are engaging right middle temporal gyrus, left inferior frontal triangularis, left middle temporal gyrus. And then our folks with stroke are showing higher engagement during scrambled than the real condition, which is maybe suggesting some sort of brain reorganization or something like that. It's pretty interesting and is not a pattern that we see in the older healthy controls, although we see it in the left angular gyrus in the young healthy controls. So interesting that it flipped from the left angular gyrus, this same pattern of scrambled over real to the right in our folks with stroke. And then I'll just move on to our last task here quickly, so I can make sure and tell you a little bit about study 4 before it's time for questions. So in the arithmetic task, young healthy controls and older healthy controls showed robust engagement of bilateral MFG. So here and here, and then again, we're seeing that again here and here in our older healthy controls. And then our folks with aphasia showing higher HBO concentration change actually during the easy relative to the hard task in left inferior frontal triangularis and supramarginal gyrus, perhaps related to some sort of processing during the easy condition, or suggesting that the hard condition may have just been too challenging for them. And we'll just kind of move on to this last study very quickly. So given that we had some confidence that we could measure task-based activation in the damaged brain using Effners, we conducted a early preliminary study with the overall aim being looking at pre to post intervention activation difference in just 5 folks with ABI who participated in the ICCR program using Effners before we had to stop recruiting folks due to COVID. So this preliminary work right here would hopefully allow us to begin to start establishing these methods for larger studies in the future. So in terms of our behavioral data, we used linear mixed effects models to assess the effect of time on overall item accuracy, similar to what we did in study 2. We looked at accuracy on the Effner's behavioral tasks by comparing the proportion of accurate responses between time points using McNamara's, and then reaction time we did linear mixed effects models. And we can get into that in more detail. This is the same processing stream that we used in study 3 with just a few differences. We did prune channels that had poor signal to noise at both pre and post time point to make sure that we were measuring the same signal from the same 2 channels at both time points, and not just seeing activation that was because a channel was active at post that was not actually being measured at pre, for example. We used more conservative motion artifact identification and stimulus rejection. And then finally, we used a session level general linear model. And I can talk about this in more detail with anybody who's interested. And what you're going to see here are T-maps for a single condition. So I'm just going to go ahead and show everybody at once. And I just want to say that these are very early results and we can't really make any strong conclusions from them, but we did see some interesting trends. So P1 was somebody who was a young man with a tumor who showed a positive response to the intervention and improvements in accuracy in conjunction with longer reaction time. And his neural results reveal higher HBO concentration change in the left middle temporal gyrus in the hard than easy addition condition, which we did not see at the pre-time point, right? So you're not seeing this activation here. So that's interesting. And then for P2, again, status post-tumor did not show a significant effect of the treatment per se, but she did demonstrate gains in accuracy in both conditions and to a statistically significant extent in the easy condition. And her activation patterns differ a lot from P1 with higher HBO concentration change in the right inferior parietal cortex in the hard and easy condition and higher HBO change in the left inferior parietal at the post-treatment time point, which is different than what we saw for her in the pre-treatment time point. And then we have P3. He was a young man who sustained a focal TBI from a gunshot wound that led to a left MCA rupture. He really presented very much like a classic left hemisphere aphasia. He did not show an overall effective treatment, but we did see a higher HBO concentration change in his right inferior parietal, right middle temporal, and left superior frontal middle in the hard than easy condition at the post-treatment time point, which appears to be an increase in activation than previously. And you can kind of see that again, I think the take-home point here is that there's, without a lot of time left, with P4 and P5, so P4 did not show an overall effective treatment, but did show these patterns in a hard versus easy addition condition with higher accuracy and a faster response time in the hard than, or in the easy than the hard task. And in terms of their brain activation, we see that in the post-treatment time point, there's higher HBO concentration change in right SFG and MFG, and then right inferior parietal middle temporal gyrus. And then finally, P5 had severe TBI, did show a positive effect of treatment with increase in accuracy in both conditions and a statistically significant effect in the easy condition. And this person shows, an increase in HBO concentration change in bilateral frontal and right inferior parietal regions in the easy then hard condition, and similar patterns that were observed in the stroke group in study 3. So this is actually kind of interesting and kind of really showing an abnormal response relative to controls in the damaged brain. So I think overall, these results have to be taken with the fact that there were a lot of channels that were pruned. And so there's a lot of missing data in these, and there's a lot more work to be done to improve this process, but at least very first early steps at trying to use Effners in this population to measure treatment-induced plasticity. And again, we're seeing a lot of variability in terms of their behavioral and neural outcomes, but more work to be done there. So I think I've already kind of given you the high levels that, we saw increases in overall cognitive function from the treatment, and then in specific cognitive domains. Effners can be used to measure language and domain general cognitive processing in healthy, younger and older adults and individuals with aphasia. And then there's very early evidence that we could probably use it to assess changes from intervention in young adults with ABI. I'm going to skip over the future directions for that work and just say that, currently I'm working as a postdoc in the lab for neuroimaging of coma and consciousness with Brian Edlow, doing a lot of fun projects. One involving looking at biomarkers of repetitive blast exposure in active duty special operation forces personnel, another looking at cognitive outcomes in white matter tracks in chronic TBI, and then another looking at cognition, participation, and quality of life in folks with moderate to severe TBI between rehab DC and year 1, and having a lot of fun doing it. And hopefully we'll be able to hear more about some of the work that's being done at MRI in the future. And with that, just a big thank you to everybody for attending today, acknowledging my funding sources, all of my previous and current lab members, and of course the folks with brain injury and all of their care partners who made all of this research possible. And I'm happy to take any questions if we have any time remaining.
Thank you, Natalie, for this very, very comprehensive program and an overview. I think we have time for one question, please.
Great.
Laurel, you'll see your hand up.
Hi, Natalie. Laurel
Hi.
Buxbaum. I don't know if we met when you were at Moss. Probably not. But anyway, thank you for your very thorough. I had a million questions, but I know there's not a lot of time.
I'm sorry. Yes, I'm happy to answer anything, even if you want to email me.
Here's a quick question. Here's a quick question. One of the challenges we often have in patient research is, and in general, a statistical challenge that we have is that when looking at two performances or two time points or one group versus another, we want to avoid the fallacy of saying that one surpassed a threshold and the other didn't surpass a threshold, and so those two are different from one another. And I thought I saw that happen a lot in, maybe I'm wrong, but you weren't very explicit about, pre versus post testing. They looked different from each other in the maps generated, but was one significantly more activated than another, so pre versus post or patient versus control? Did you do those kinds of comparisons? There was both in study 3 and study 4, those questions were raised.
Yeah, so that's a very good question. So essentially, were they performing similarly to one another? Like,
not just the performance, I'm even talking about in the Effner's data, where you have, one looks like it's activated the IFG and the other didn't activate the IFG. Well, there are thresholding issues there. There's significance thresholds. You have a color map that shows how significant one, let's say, I'll make it up, but let's say your control showed IFG was bright red, but the patients were yellow.
Yeah,
Okay, so you said, oh, they didn't surpass the threshold, the patients.
right. Yeah. No, I'm
But
happy
what
to
you
show
really
one of
want to
those.
know, I mean, as we know, you can have, one can be just above a statistical threshold and one can be just below, but they may not be different from each other. So
Right.
if you haven't done that, I urge you to try to take that approach with your data, because it would enable
Right.
much stronger conclusions.
Oh, thank you. Yeah. So I will say these here are not actually T maps, and so these are actually showing, this is you can actually compare the color bars here, because these are they're all using the same color bar. So if it's red here, it's the same red in the.
Right, but that doesn't mean, so if one's red and the other's orange, doesn't mean one's more than another.
That's true, because they weren't pitted against one another specifically.
Exactly.
Yeah. Yes,
Okay.
that's a good point. Yeah, thank you.
Okay.
Thank you for that. Yeah, much appreciated.
Thank you so much, Natalie. Unfortunately, we're quite out of time, and the people in the clinic need to return.
Oh, totally
But
fine.
it was
Yeah.
really terrific to hear from you and to learn what you've been up to. Thank you so very
Great.
much.
Well, thank you very much for the invitation and for the comments as well. Thank you.
Thank you.
Thanks,
Bye,
Laura.
bye.
Okay, bye-bye.
Bye. Bye.
Bye-bye. Take care.